All right, hello everybody. Today we are going to do a DNA fingerprinting simulation. I've got all my equipment right here. I'll pause it at some point and I'll bring us over to the DNA electrophoresis machine. Let me show you some of the equipment that we have. Right here we have the micropipette. Oop, it's upside down for you. There we go. Let's try that. And notice that I can turn the dial on the side. That sets how many microliters I'm going to suck up. Today we want to be sucking up 25 microliters of fluid. Now, we're not allowed to use real DNA, so we're going to use simulated DNA. It's just kind of different colors. So I've got green, blue, what are my colors here? Red, purple, orange, and brown. So I've got some fun colors here that we're going to be working with. And then just to remind you with the micropipette, you can add a sterile tip every time. So we want a new tip every time we get a new color so that we don't contaminate it with DNA. It's not real DNA, it's fake DNA, but whatever. Okay, so watch what I'm going to do. I'm going to go ahead and place a tip onto the micropipetter. I'm going to choose one of my colors. So I'm going to choose the color green first. So I'm going to open up the green. Feel free to fast forward if you want to while you're watching this. I'm going to suck up the green, so I put it tip all the way down to the bottom and I release it very slowly. And then I've got green in the tip right here. And then I go down here and I add the green into the gel. And I'll show it to you in just a second. And I remove it. And now if I show you the gel, I can show it to you this way. You can see there's green in there. But I can also show it to you this way. Can you see there's green in this well? There's a little divot in there, and I've added the green color to it. So now I'm going to keep adding different colors. So I'm going to go for the next one is blue. Oh, by the way, while I'm doing this, I want you to record what color we're putting in each well. So the first well that we did was this one right here. So you're going to put green in there. I wonder if it's backwards. I feel like the image is flipped around. So the very furthest left well, that's the one that you're going to make green. I'm going to go ahead and color it green with my colored pencil. If you're doing this on Cami, you can color it green. All right, the next one, I'm going to pop off this tip because it's contaminated. So that one's gone. I'm going to put on a new sterile tip, and I'm going to get blue. And I'll suck it up nice and slowly because it's really thick. And I'll put it back in here, and I'll eject it in. Keep going, get all of it in there. A little bit came out the top. There we go. So I'm ejecting my tip, and you can see that I've now added blue to that one. Next, I'm going, and so color in blue next. I should do that so I don't forget what color I'm doing. So I'm going to color in blue on my paper. The next one I am adding is red. I better get a new tip. There's my tip, and I'm going to add red. And I suck it all the way up. I hope I got a good one there. And add red in. This one is leaking, unfortunately. This micropipette isn't working very well. I'm going to suck up a little bit more color because the tip is leaking. I did not get the best one. There we go. Now we got plenty of color in it. Show you right here. You can see that now we've added red. So color that next line red. And I shot that over me, making the next one red. After that, we're doing purple. I'm going to suck up the purple. Do it nice and slow. Make sure that I've got enough in there. And I'm injecting it all in. There's the purple. And I'm going to color it purple here. Next is orange. Going to get rid of the tip. Oops. Get rid of the tip. I'm going to add a new tip. Suck up the orange. Do it nice and slowly. I'm now going to add it here, even though it's leaking a little bit. Well, it can make it work. Here we go. That's the orange. Wipe it off a little bit. That leaking is a pain. And I'm ejecting the tip. Ejecting the tip is kind of enjoyable. You can see the orange color now that I've added. So I just have one left. I'm going to add my orange colored pencil onto the paper here. And then the very last one is the lovely color brown. I always do brown because it makes the prettiest isn't the right word, makes the most interesting fingerprint. So again, this isn't real DNA that we're working with because we don't have DNA accessible. What we're working with is color molecules, but it separates super, super cool. I'm going to add a little bit more because that one just is leaking so much that it didn't add very well. And I hope I got it. Okay, so that's that. I'm going to eject this tip and close these all up. Now I'm ready to take my gel and I'm going to go put it in the gel electrophoresis machine. I colored in the brown. So you can see I've colored in the colors that I did. So make sure you have those right colors. If you're doing this on Cami, add those colors. All right, I'm going to pause this for just a second. I'm going to walk over to the electrophoresis machine. All right, this is the electrophoresis machine. I'm going to fold this over. Hold on, this is going to be a little bit awkward. There we go. And I'm going to show you, oh, that's just not working at all. Okay, hold on just a second. I was hoping I could show it to you. I thought if I folded it over. This does not have a camera on the back. Oh, I know, it does have another camera. I don't know how to show you the other camera. Okay, so I'm just going to have to do this a little bit awkwardly. This is the machine. It's called an electrophoresis machine. And we put this is the gel that I just did. It's right here. You can see all the vibrant colors in the one that I just did. So right here. This is the positive end up here. And down at the bottom is the negative end. And we're going to add electricity to it. Now, there are some gels that are already placed in this electrophoresis machine. They're old and the colors are fading, so they're not the one I'm doing. But this is the one I'm working on right now. So give me just a second. I'm going to attach the lid. See if you can watch me attach the lid. So again, the red indicates the positive end, and the black indicates the negative end. I have to get that on really nice and tightly and make sure, yep, it's all good to go. So now I'm going to turn on the switch and that adds the electricity. So now the negative charge is on this end and the positive charge is on this end. You might be able to see if I can get the camera good. Can you see that there's little bubbles rising up on the negative end from the wire that goes across there? So that shows me that it's actually working. And now I'm going to wait about 20 minutes and we'll develop it in 20 minutes and take a look. I'll pause it and I'll come back in 20 minutes. All right, I'm back already. Seems like no time has gone by, right? All right, I'm going to go ahead and turn the machine off. So I'm flipping that off. And this is what it looks like now. It's all kind of got condensation on it. I'm going to take the lid off, which I might not be able to do. I'm doing it with one hand, okay? Nope. I need two hands for this. There we go. And remember that there are additional gels in there, but this gel that's on the far upper left-hand corner, that's our gel. And now we're going to analyze it, and we're going to see what all those different lines mean and everything. So let me take it out, and I'm going to carry it into the other room, and we're going to take a look and see what it all means. Give me just a second. I'm going where my colored pencils are. Okay. So let's take a second to really look at what the DNA fingerprint looks like. So I'm going to hold it up to the camera so that you can see it a little bit better. I'm going to make sure that it doesn't drip on my computer. That would be bad if it dripped on my computer. Can you see? There we go. I'm trying to hold white behind it so you can kind of see. Okay, so what I want you to notice is the color of the lines. There's only 3 colors present. There's no purple. There's no green. There's only blue, red, and yellow. Well, what happened to our purple? What happened to our green? What happened to our brown? Where did they go? Well, it turns out, you guys, that I made those colors by mixing together the colors red, yellow, and blue. So I had 3 colors, and so the red was just red all by itself, but brown was a mixture of red, blue, and yellow. So let's go ahead and take a look at our paper. We're going to color it in, and then we're going to answer the questions at the bottom. So the first one that we looked at was green, and so it might be really hard for you to see, but green is this one right here, right there, and it's got a band of blue and yellow. I'm sure you can see the blue. You might have trouble seeing the yellow. So I'm going to draw, the only colors I'm going to draw with now are blue, yellow, and red. I'm not going to draw with these other colors because these colors don't really exist anymore. They broke into their colors that what the artists call the primary colors. So I'm only coloring with blue, red, and yellow. So for this first one, I'm going to make a blue box, a band for how far the blue traveled. And then quite a bit above it, I'm going to make a yellow band. So there's like a gap between the two. So it looks like this. There's my blue band and there's my yellow band. I'm kind of making a cartoon of what I saw on the gel. I'm not drawing it exactly what it looks like, just drawing a cartoon. There's the blue band, there's the yellow band. All right, the next one is pretty easy because it was just the color blue. So that's this one here. And all it has is a blue band. So what's important is that the blue travels the same distance every time. So I added my blue color and my blue band. Notice it traveled the same distance as it did in the green one. All right, the next one was red. So let's take a look at the red. Can you see the red? It's the third one, and the red is just a red band all by itself, but it traveled further than the blue did. So I'm going to make the red band a little bit higher than the blue band was. And there's nothing else to the red. It just broke into its original red color, and that's it. All right, purple. Can you guess what purple is? This purple one's a little bit hard to see, so it's this one right here, but it's got a band of blue and a band of red right on top of it. So I'm going to go ahead and color a band of blue down low. It's at the blue traveled the same distance that all the other blues did. And then I'm going to color a red band on top of it that traveled the same distance as the red before. So here's my blue and red. Notice the blue is traveling the same distance every time, and now the red has traveled a little bit further. That tells us something. Remember, how far they're traveling tells us something. All right, the next one, again, this is hard to do on this particular, like on a video, it's hard to show, but the next one was orange. So that's our second to last one. And there's a band of red, and then on top of it, there's a band of yellow. So I'm going to show the band of red at the same height that all the other reds were because they traveled, all the red travels the same distance. And then I'm going to draw a band of yellow on top of it. So let's see if you can see that. So there's my orange. There's the red went the same distance as all the other reds did. And then the yellow went the furthest distance of all. Okay, and then the last column is the brown. And this one is also hard to see. But in the brown, I have a band of blue, red, and yellow on top of it. Guess how I made the color brown, everyone? I mixed a little bit of blue, a little bit of red, and a little bit of yellow. So when you draw this, I want you to make the colors travel the same distance every time. So this is what brown looks like. Blue is at the bottom, red is in the middle, yellow's on the top, and notice that the yellows are all the same height, the reds are all the same height, and the blues are all the same height. All right, so now let's get down to the bottom and decide what the answers are to the questions. So question number 3 says, use colored pencils to draw in blah, blah, blah. We already did that. Question number 3 says, if opposite charges attract, I'm showing this here, if opposite charges attract and like charges repel, what was the charge of the color molecules? Well, there's negative charge down here and there's a positive charge up here. So if the like charges repel away and they're being attracted to the opposite charge, then that must mean that these guys originally were negative and they were repelled by the negative charge down at the bottom and they're moving closer and closer to the positive charge. So you should write that they were negative. All right, next, question number 4. Which color molecule was largest? Well, remember, the big molecules have trouble passing through the gel, and so they don't travel as far. So the big molecules are blue. Blue is the largest color molecule. It moved slowly and not as far. So how did you know the answer to number 4? Blue did not travel as far as red or yellow. Blue did not travel as far as red or yellow. All right, question number 5 or 6. Using the same logic, which color molecule was the smallest? Well, the small ones can travel really fast across the gel. So clearly our smallest one was yellow. It was the smallest. Red was medium and blue was the biggest. So you would say yellow. And now for the very last question, why did some colors produce more than one line? Let me ask you this one. Why did the brown have a blue, a red, and a yellow? Hopefully you're thinking of the answer. The reason that some of them produced more than one line is that they are a mixture of colors. So I mixed 3 colors to make brown, and now they split into their 3 original primary colors. All right, ladies and gentlemen, that's it. Let me know if you have any questions. Bye-bye.